Good morning, respected teacher and my dear friends. Today we are going to present on the topic database searching application in computational biology and chemistry. This presentation explains how database searching works and why it is important in bioinformatics and chemistry, especially for handling large biological data. Before starting, I would like to introduce our team members. First, myself, Sheikh Sabbir Ahmed Alif, ID is 253-15131. Next is Maria Hossein, then Abu Sain, then MD Abirul Islam Pritam, and finally MD Zakaria Firdos. Now I will start our presentation with the basic concept of database searching. Slide. In this slide, I will talk about what database searching is. Database searching means find specific information from a large collection of data, which we call a database. In computing, it uses different techniques like indexing and query processing so that data can be found quickly and accurately. In bioinformatics and chemistry, database searching is very important because scientists need to search DNA sequence, protein structure, or chemical compounds. Without database searching, it would be almost impossible to handle such a huge amount of biological data. Slide change. Why is critical? Now let me explain why database searching is so critical. Database searching work as a bridge between computer science and biology. Today, biological data like and molecular structure are going very fast. This creates a big data problem. Biological cannot manually search millions of records. That's why they need computer science algorithm to do this work efficiently. The main goal of database searching is to convert raw data into useful knowledge such as identifying a gene or discover a drug target. So database searching plays a key role in modern biological and chemical research. Slide change. Classification of biological database. In this slide, I will briefly explain this classification of biological database. First, we have primary database. This database store raw experiment data submitted directly by research. Example include gene bank for sequence and PDB for protein structure. Second, there are secondary database. This database contain and data that comes from primary database. Example are PFM and ProSite. Finally, we have composite database. This database combine data and multiple source and remove uh, redundant. Example include OWL and NR database. This qualification helps researchers understand where the data comes from and how to is used. This is all of my side for this slide. Now I would like to hand over the presentation to the next speaker who will explain the following slide in more details. Thank you.
My name is Maria Hussein. My ID number is 253-15170. Today I am going to start with the biological database, the main sources where biological information is stored and shared. First, we have NCBI, which stands for National Center for Biotechnology Information. It's often called the Google of biological data because it provides access to many databases, including GenBank for genetic sequences and PubMed for scientific papers. Researchers around the world use NCBI to find and analyze biological information. Next is UniProt or Universal Protein Resource. This is the main database for protein sequence and functional information. If scientists want to know what a protein does or how it is structured, they usually go to UniProt. Finally, there is PDB, the Protein Data Bank. It is the main archive for 3D structural data of large biological molecules such as proteins and nucleic acids. PDB helps scientists visualize molecular structures and understand how they function in our body. So, these three NCBI, UniProt, and PBD are essential tools for biological research. Now, let's move to chemical databases, which fall under the field called cheminformatics. The first one is PubChem. It's a very large database that contains information about chemical molecules and how they and how they behave in biological tests or assays. It helps researchers understand chemical properties and activities. The second one is ChemBall, which is a manually created database of bioactive molecules with drug-like properties. It's mainly used in drugs discovery to find potential compounds for medicines. When searching in these databases, scientists don't just use text. They can search by molecular weight, chemical structure, or functional groups. These advanced search options make it easier to find specific chemicals or molecules based on their features. Finally, let's discuss text-based search methods. This is the simplest and most common way to find information in biological or chemical databases. In this method, we use keywords, accession numbers, or author names to retrieve data. For example, if we type a gene name in NCBL or PubMed, we get all the related information and research papers. We can also use Boolean operators like and, or, and not to refine our search. For instance, cancer and protein will show results that include both terms, while cancer not virus will exclude virus related studies. Text based searching is quick, effective, and forms the foundation of all other search techniques. That's all about my topic. Thank you for your attention.
Hey everyone, my name is Mohammed Hossain. My ID is 2531517. I'll be discussing about the slide sequence alignment, which is one of the most important concepts in bioinformatics. The main problem is that text-based searching doesn't work when we want to find biological similarities. In real life, DNA and protein sequence change over time because of mutation and evolution. So instead of matching text, we compare sequence. In sequence alignment, a DNA or protein sequence is compared to millions of sequences in a database to find similar regions. If two sequences are very similar, it usually means they are related in terms of function, structure, or evolution. This helps scientists identify genes, predict protein function, and understand diseases. That's why sequence alignment is called the core of bioinformatics. Moving on to the next slide, the BLAST algorithm. Next slide, please. Okay. To perform sequence alignment efficiently, we use an algorithm called BLAST, which stands for Basic Local Alignment Search Tool. BLAST is the most widely used tool for searching biological sequence. The reason BLAST is so popular is because it is fast. Instead of comparing very single base pair, it uses a heuristic approach, which means it finds very good matches quickly. BLAST works in three simple steps. It breaks the query sequence into small pieces called words. It searches the database for matching words, then it extended those matches to find longer algorithms. Because of this smart method, BLAST can search huge database in seconds. Moving on to the next slide, types of BLAST searches. Next slide, please. Okay. There are different types of BLAST depending on what we want to search. BLAST N compares DNA sequence with DNA database. BLAST P compares a protein sequence with a protein database. BLAST X translates a DNA sequence and compares a protein, which is useful for finding coding regions. T-BLAST N compares the protein sequence against translated DNA sequence. Each type is used for a specific biological problem, making BLAST a very flexible and powerful tool. So overall, sequence alignment and BLAST play a crucial role in connecting computer science algorithm with biological research. Thank you, everyone.
Thank you, Sam. I am MD Abirul Islam Pritam, ID 253-15198. From here, I will continue this presentation. Up on the search method we have just discussed, I'd like to take us deeper into the technical engine that makes these biological searches possible. We are going to look at how computers actually grade a match, how we look beyond single text sequences, and finally the algorithms running under the hood. First, discuss about fundamental challenge. How do we judge a good match? When we look at two biological sequences, our eyes might spot similarities instantly, but a computer needs a mathematical framework to quantify that similarity. This is where scoring matrices come. Primarily, we use substitution matrices like PEM and Blossom. Think of this as a point scoring system. If two sequences share an amino acid that is critical for survival and therefore conserved by evolution, the matrix awards a high possibility score. If there is a random mutation that is unlikely to happen naturally, the matrix appears a penalty. But a sin alone isn't enough. We need to know if that score is statistically significant. That is why we use the e-value or expect value. This tells us the number of hits we could expect to see simply by random chance. However, sequence similarities isn't the whole picture. This brings us to structural searching. There are cases where the search's similarity is very low, meaning the text doesn't look alike, but the 3D shape of the protein is almost identical in biology. This is where structure searching is important. To find these hidden matches, we moved beyond 1D sequences and use tools like VAST Vector Alignment Search Tool. Instead of matching letters, VAST looks for genetics similarities in the protein data. We can see under the hood at the algorithm that powers these massive searches. We are dealing with gigabytes of generic data, so efficiency is the key. Now we talk about suffix tree. These are used for incredibly fast string matching. They allow us to find specific modifiers or pattern inside massive genome databases almost instantly. Then another thing is a hash table. This is the secret weapon behind the BLAST algorithm. Instead of reading every letter, it uses hash tables for constant time lookups. It breaks sequences into small words or k-tuples to find quick initial matches. And the most important thing is dynamic programming. When we need absolute precision, we turn to algorithms like Needleman Wunsch and Smith Waterman. These provide mathematically optimal alignments. While they are the most accurate, they have a higher time complexity of 0s, making them slower than BLAST but essential for detailed analysis. From the databases, we query to the scoring matrices and algorithms that process them. Computational biology allows us to run raw data into meaningful biological discovery. Thank you for your attention. Now, I would like to hand over the presentation to the next speaker, who will explain the following slides in more detail.
Assalamu alaikum. My name is Jakari Firdos. My ID is 253152200. Now I'll talk about the challenges in database searching. First challenge is data heterogeneity. Different database use different formats like FASTA, XML, and flat files. Because of this searching, all database together become difficult. The second challenge is noise and errors. Experimental data is not always perfect. If the data contains error, the search result may be wrong or misleading. The third challenge is redundancy. Sometimes the same biological data appears many times in database. This wastes time and computational resource and affects search accuracy. So moving to the next slide. The next slide is the future trends of database searching. First is cloud computing. Large biological databases are moving to the cloud. This helps in faster and distributed searching. Second is AI and machine learning. Deep learning models like AlphaFold help predict protein structures. AI can find patterns that traditional algorithms cannot detect. Third is federated search. With federated search, we can search multiple databases at the same time using a single interface. This saves time and effort. The next slide is conclusion. Finally, I want to conclude my presentation. Database searching is a fundamental tool in modern biology and chemistry. It depends on computer science concepts like efficient indexing, heuristic algorithms such as BLAST and large storage system. As CSE student, understanding this database helps us build better tools for drug discovery and genetic research. Thank you everyone for listening.